I am Tushar V from Telecom Department, and I am going to explain you about the abstract of our topic. Nano manipulation inside a scanning electron microscope has been employed to maneuver and characterize nanomaterials. In the scanning electron microscope, many studies require micro manipulation tools that can be flexibly and comfortably used for the handling and positioning of objects ranging from few millimeters down to fractions of microns. The scanning electron microscope presents an automatic alignment procedure for a four source photometric stereo technique for reconstructing the depth map in the scanning electron microscope.
Hello, myself Mohammad Rehan Qureshi. I am going to give the introduction of our topic. The scanning electron microscope instrument is made up of two main components, the electronic consoles and the electronic column. The electronic consoles provide control knobs and switches that allows for instrument adjustments such as filament current, accelerating voltage, focus, magnification, brightness, and contrast. The FEI Quanta 200 is a state-of-the-art electron microscope that uses a computer system in conjugation with the electronic console, making it unnecessary to have bulky console that house control knobs, CRTs, and an image capture device. All the primary controls are accessed through the computer system using the mouse and keyboard. The user need only be familiar with the GUI or software that controls the instrument rather than control knobs and switches typically found on older style scanning electron microscopes. The image that is produced by SEM is usually viewed on CRTs located on the electronic console, but instead with FEI, the image can be seen on the computer monitor. Images that are captured can be saved in digital format or printed directly.
Good morning, sir. My name is Ishan and I will be doing literature survey. De Broglie introduced the concept of corpuscle wave that led to the development of the wave electronics optics and thus steered the scientists towards electron microscopy. In 1897, Thomson discovered electron, and in the same year, discovery of magnetic and electric field as lenses and wave nature of electrons took place. Max Knoll and Helmut Ruska invented first commercial transmission electron microscope having 10 nanometer resolution, which proved year by year to 1 nanometer and eventually to 0.2 nanometer. Then in 1965, British-born American scientist Albert Victor Crew added a field emission gun in transmitting electron microscope, which raised the resolution further to 0.05 nanometer. Moving on towards the problem statement. The problem of optical microscopy is about limited resolution because we are working in a band of wavelength which are very large, which leads to greater diffraction, and the microscope can't produce the image of an object that is smaller than wavelength of light wave. Optical microscopy does not provide 3D structure, whereas scanning electron microscope provides us digital 3D image. X-rays are able to penetrate a sample because of their high energies and extremely low wavelength. That's why it does not provide the surface characteristics and it could damage biological sample. Now, motivation. Why did we choose scanning electron microscope as our topic? Our interest in the field of scanning electron microscope and fascination on the topic was the reason for which we have chosen this topic. To understand, analyze and apply the topic which were taught in our senior secondary classes. SEM plays a crucial role in the production of microchips, and this was the other reason for choosing this topic, as India is leading a very high position in the form of electronics and microchip design nowadays. Now, what are our objectives on this? To study the topography of materials for quality control, electrical properties, and failure analysis by knowing the type of arrangement of constituent particles, that is, the crystal lattice. Another, three-dimensional capacity of SEM is valuable to microchip design and production in order to increase the accuracy in criminal and forensic investigation like jewelry examination, handwriting, and fingerprint analysis. Fourth, medical science field like understanding the morphology of the virus and bacteria. Fifth, also it is used in soil quality and toxicity measurement for farming.
My name is Harish. I will be handling the methodology. Let us see the comparison of transmission of electron microscope and the scanning electron microscope. Transmission electron microscope used to study the ultrastructure of the cell. Scanning electron microscope used to produce the excellent image of the surface if the cells. Transmitted electrons is used as a source in the transmission electron microscope, and the scattered electrons is used as a source in the scanning electron microscope. Moving on to the next point. Methodology. The automated nano manipulation inside the SEM. A nano manipulation system is integrated to the scanning electron microscope by mounting a head assembly onto the specimen stage of SEM. The fine positioning unit contains the piezoelectric tube having travel ranges of 10 micrometer along the axis of the tube and the 100 micrometer along each of the two transverse direction with 5 nanometer nominal resolution. Point B. Recognition of the probes and the nanowires. When the 4 probes and the 1 or the multiple nanowires are in the field of the view, they are visually recognized and identified. The probes and nanowires are distinguished by comparing the area surrounded by the counters. Since the nanowires have a smaller areas, the 4 probes are distinguished from each other by comparing the position of the centroids out of the counters, and the positions of their tips are determined as either the highest or to the lowest probe counter. Feature tracking. The visual tracking provides the motion, positioning information of the probe in the image frame as the position feedback or the version based contact detection or visual servo control. The system first conducts the visual recognition in a frame of the image 1 to obtain the coordinate x1 and y1 of the probe tip. The rectangular patch of the image containing the probe tip is recorded and templated. Intelligent tweezers, a teleoperation. Numerous experiments and studies in a various research fields going on as present required to manipulation of the object smaller than 100 micrometer. These experiments often have performed in the SEM due to the tiny dimensions of those objects, which are usually also fragile and hard to handle. Flexible mobile macro robots are helpful tools in such situations. Compare bending behavior and piezolegs. The legs are the tube-shaped piezoelectric actuators. The outer electrode of these tubes are divided into four parts. The vector addition for this compensation is shown in a resulting in a corrected vector. Gear change. In order to proceed towards a smaller and smaller object, the probe to be handled or to the nano positioning tasks are described in chapter 3. As the next lower gear can be chosen. The setup modeling. The positioning platform is mounted inside the SEM vacuum chamber and controlled using the 3-channel piezo controller. The maximum possible motion on all axes up to 100 micrometer with a resolution of 2 nanometer. The primary computer. Automated sequence, initial setup. The subject is framed and magnified and focused as a normal optimum scanning electron microscope imaging. Illumination models. Rest of the all proposed a radial model for the illumination. This is motivated by observation that the electron beam is radially symmetric in nature.
So finally, moving towards the result and conclusion of our topic. A method for vision based contact detection was developed to detect the contact contact between a probe and a subtract for determining relative vertical positions of the probe's tips and nano objects to be manipulated. The development of the micro robot acting as a pair of tweezers for handling tiny objects in the scanning electron microscope has been presented. For the enhancement of the control system, a closed loop control has been proposed basing on a sensor system that mainly consists of a standard scanning electron microscope. A visual servo control system with a feed-forward controller was built for closed-loop control for multiple nano manipulators, which demonstrated significant improvements in response time in comparison to feed-forward controller-free scenario.